Now it's my pleasure to introduce Dr. Wendy Chung. Wendy serves as the principal investigator of the Simons VIP project, which characterizes behavior, brain structure, and function in participants with genetic copy number variants, such as those at 16p11.2, that are believed to play a role in autism. She joined the Simons Foundation in 2012 as Director of Clinical Research after serving on Safari's Scientific Advisory Board. Wendy's a clinical and molecular geneticist. She's currently the Herbert Irving Associate Professor of Pediatrics and Medicine at Columbia, where she directs the clinical genetics program and performs human genetics research. She clinically assesses children with autism spectrum disorders and intellectual disabilities and uses advanced genomic diagnostic methods, including whole exome sequencing, to identify the underlying genetic basis for neurological conditions. In her work, Wendy strives to facilitate the integration of genetic medicine into all areas of healthcare in a medically, scientifically, and ethically sound, accessible, and cost-effective manner. We've had the honor of getting to know Wendy over the past year, and we very much appreciate her support and the wisdom she's brought to every discussion. Please join me in welcoming Dr. Wendy Chung.
Thanks, Dee Dee, and thanks for having me here today. So I'm going to tell you about a project that we've been doing for the last 2 years, and with many of the folks actually here in the audience today. I'll give shouts out to people that have been working with us at the Boston site. And it's been really, I think, quite illustrating, as you'll see in terms of some of the things that Jordan Smoller was talking about, some of the boundaries and sort of the lack of distinction in terms of diagnoses and going upon that journey. So as many of you know, when we think of autism, it's really not autism, it's the autisms, because it's really a group of disorders that have some shared and overlapping features. When we ask the question, though, of what causes the autisms, it's really obviously not a single etiology. With recent technologies, as Dr. Smoller was talking about, there's been some ability to try and elucidate some of those underlying genetic and genomic etiologies, but really we're still quite far from understanding for most individuals what might have caused their issues with ASD. So as we've been doing this, and the Simons Foundation, as some of you know, has sponsored something called the Simons Simplex Collection that we did to take singleton cases, that is individuals that didn't have a family history but just had one child with autism, to try and understand what that underlying genetic basis might be. And when you start looking at cases like that or looking at autism in general, you realize that it's quite a complex disorder. There are certain types of genetic etiologies that are these de novo events, these rare de novo events, or copy number variants. And as we started doing that in the early days by using chromosome microarrays to look at those copy number variants, the one that we saw that was most commonly seen within the Simons Simplex collection was actually 16p11.2. And even though I say it was the most common, to be quite modest about this, this still only accounted for just less than 1% of all individuals with autism. In that case, we saw that both deletions or duplications of this 600 kilobase interval could be associated with autism. And so this is one of these what we call canonical copy number variants. That is that it's the exact same interval that we see in all those individuals, encompasses a total of 29 genes in this particular case that are either one less copy, so a total of 1 copy instead of 2, or 1 extra copy, a deletion, or 3 copies total instead of the normal 2 copies. And so realizing that this was a relatively common cause of ASD, we decided to take a strategy where we would start with the genetics and then take it where it led us, hoping, of course, that this would help us to understand in a more well-defined subset of individuals with autism some of the following features. Trying to understand for those individuals the composite and the complex relationship between some of the behavioral issues, the neurological issues, the psychiatric issues, as well as how that related to some of the medical issues, the comorbidities. To understand in many cases, not just at a behavioral level, but actually at a structure and a functional level of the brain, being able to do advanced imaging methodology to try and understand the correlations between what we could see behaviorally was going on with structurally and functionally what was going on in the brain. And importantly, to be able to actually see how this evolved, to be able to follow these individuals as they were growing up longitudinally to see how this evolved, how this changed, how it might respond to different treatments or different therapies. And of course, ultimately, ultimately to be able to understand how this might be more broadly applicable to other individuals with other forms of autism. But again, we started out with the genes in this particular case, hoping that that might give a more homogeneous group, a more well-defined group that might be more alike with each other, that hopefully we would be able to eliminate some of the noise that we sometimes see with our studies when we have just a sort of garden variety forms of autism. As we do this, this was really facilitated, and I'm just going to give an unabashed advertisement, but this was facilitated really because at the same time there was now a clinically available test for a chromosome microarray so that patients with autism could actually get this diagnosis. That was really critical to being able to do this study. And so the reason I said I'm going to put out an unabashed advertisement is for any of the families out there. I will tell you that even though it may have been several years since if ever, that your child saw a geneticist, for instance, now might be a good time to actually go and get an update or talk with any of your providers about an update, because there now really is improved testing to be able to subdefine the different type of autism or the etiology for the autisms. And increasingly, I do have the faith that it's going to be helpful to understand exactly what caused the autism, because hopefully we'll be able to provide better prognostic information, more specific information that you might be able to use for your child. So as we did this, again, this was facilitated because now families knew that their particular child had, for instance, a 16p deletion or duplication, and we realized that many parents are really quite savvy, quite motivated to be and wanting to participate in research, and we had to simply give them the opportunity. So we set this up, and for any of you that might want to replicate this, I can tell you it's not all that hard to do. We set up a website. We paid Google ads so that if you type in 16p11.2, you'll see popping up on the right-hand side, Simon's VIP Connect. And families, we wanted to be able to actually reciprocally help them with what they were dealing with, at the same time going on this journey with them and learning from them. So with this, we actually embedded and we curate and we maintain a lot of good information for families that's specific to 16p11.2. We have webinars, we have an annual family meeting. We're trying to actually, as I said, as we learn, give that information back to families, but realizing that we desperately need to partner with them to learn, that they obviously are critical to our understanding for this. So parents register with this. Just to give you an idea, as we started this out literally 2 years ago, or just over 2 years ago, this just shows you how rapidly we accumulated families, and these are actually families from around the world. As we've been doing these, approximately 8 new families registered per week, and these were really highly motivated individuals to do this, and it was very nice for us to also be able to supplement this. Besides the online community, we also have an 800 number for families that may not be so savvy in terms of internet connections. And we have ways for families really of any type of means or educational background or access to be able to register with us. And this line keeps going up almost exactly at the same rate, just to show you that more individuals are getting diagnosed, more individuals are finding out about this, and there's a very large number of individuals for what still is a very rare disorder to be able to participate in these types of studies. This is just a map of the United States, but we actually have around the world families registered with us. We actually have very strong collaborators also in Europe that are doing similar parallel studies that we've synergized with, and just to be able to show you again that this is something that ends up being relatively widespread. As we did this, we wanted to be able to make sure families could get access to our research studies that I'm going to be describing to you. So we set up regional centers of excellence. The one here is at Boston Children's. We've got one in Baylor. We've got one out at the University of Washington. And then as I'll show you, we also had some imaging sites at Children's Hospital of Philadelphia and then at UCSF. But again, we tried to set this up to be accessible to families to minimize the travel burden for them. As we've done this, our original goal was to collect 100 individuals with the deletion and 100 individuals with the duplication, knowing that that was the start, but that was a reasonable start to try and be able to characterize what the phenotype or what the associated clinical features were, and to realize that as we were doing this cross-sectionally originally, we would get individuals of all ages, some little ones, some older ones, some adults even. And just to give you an idea of the neurological features that we saw with this, with the deletion carriers on this first column over here on the left, and the duplication carriers on the right, we saw that this was by parent report at this point, as well as medical history review. We realized that many individuals have many of the same features that we commonly see in our patients with ASD. Many of them had problems with being hypotonic. Parents would describe that they were awkward, they were clumsy in terms of their coordination or their gross motor movement. And interestingly, and I'll come back to this again, one of the things that we noted was this mirror or this reciprocal phenotype that we were seeing in terms of deletion carriers having relative, if not absolute, macrocephaly, where conversely, the duplication carriers having either absolute or relative microcephaly. That is, it seemed that that copy number or that dosage seemed to be going with head size or head growth. We also had not an insignificant number of individuals with fraction of individuals with seizures, and I'll show you a little bit more data about that. And then interestingly enough, and this was something that was already known, children with the deletions in particular, as they were getting older, were having trouble with their weight, problems with obesity. This was not simply related to any of the medications they were on. This was a primary and fundamental part of the disorder that we were seeing. So just to describe to you how this worked, again, we had centered at Geisinger and Emory our recruitment to be able to initially get folks in the door. Then, as I said, this phenotyping or these clinical assessment cores. I'll just describe for you, this was no easy task for our families. I really applaud them for their great dedication to this. They would spend as many as 2 or 3 days with us at one of these clinical assessments. Ellen Hansen, who's here, led the team at Boston Children's and had a fabulous team to be able to patiently be able to nurture and guide the families through this, because as you can imagine, many of these tests and being able to sit in an MRI scanner and being able to go through lots of questionnaires can be enduring for families, but they were real troopers in terms of getting all of this back. An important part that we built into this, and I really applaud our psychology team for this, is we wanted to again give back to families. And just as a sort of tip that I'll give to researchers, I think this was a really important thing. They actually got comprehensive evaluations that we had provided for their children that was useful in terms of their school, their feedback of their educational programs. So it was, I think, a very valuable experience reciprocally for both the research teams as well as for the families. And then finally, for a certain number of individuals who were able to, we put them through some very sophisticated functional imaging, functional MRI, MEG, where they went to a second site, as I said, at CHOP or UCSF. And I won't have time to show those data today, but that's actually a lot of interesting functional data in terms of what we're developing now. This is just to give you a flavor of this. This is literally the battery of different assessments that we put the individuals through. Many of them were to get a very rigorous evaluation in terms of the diagnosis for ASD, so going through an ADOS and ADI, being able to go through a neurocognitive battery, including IQ testing, the Vineland, being able to look at repetitive behaviors, being able to assess language, because as I'll be showing you, language disturbances is a very significant part of the disability for this particular copy number variant, and being also interested in what the family was going through, so being able to assess parental stress within this, and as I said, even features in terms of neurological status, very primary things in terms of motor function. So really quite a comprehensive evaluation. I can always say there were things we added as time went on and we learned more about this and defined it, but really a very rich battery in terms of understanding this. Just to give you a sense, the next few slides I'm going to show you are actually for our first 101 deletion carriers. As you can see, it was divided, about a quarter of those children, the little bitty ones, less than 5 years of age, but many of them older, greater than 5 years old, where we really felt that we could get a good assessment in terms of autism and be able to have stable diagnoses. But overall, the average age of our children in that study, about 10 years of age. So here's one of the points, and you'll be able to see that this is a theme that's going to come across this. As we did this, we tested not only the children with the deletion or duplication, but we thought it was very important to actually include their other family members. So we had family controls in this that genetically we knew either did or did not have the deletion or duplication. And we included within that in blue here the parents, in green here the siblings, again that did not have the deletion in this case, as well as the individuals that we call the probands, or the initially identified child with the deletion, and this is IQ, full-scale IQ along the x-axis here. And so one of the things that you may not be surprised by, but in fact when you look at the parents, for instance, or the siblings, 100 again being the average in terms of the population average of an IQ, we have a relatively higher IQ population. These are individuals who have been sophisticated enough to join our study, to be able to navigate themselves to get to one of our study sites, to put up with us for 2 or 3 days. And so in doing this, we have a very sort of higher than average, probably, group of parents. But that what you can see is for our probands, again with the deletion, they're clearly shifted over to the left. You can still see that there's a normal distribution in those individuals in terms of their IQ, but they still are shifted approximately 2 standard deviations, 1.8 standard deviations to the left and following a similar curve to their parents, just leftward shifted. Now, interestingly, if you take as 70, the sort of dividing line in terms of individuals with intellectual disabilities, for instance, that being 2 standard deviations below the mean, you can see that most of these individuals, by definition, are not intellectually disabled in terms of the definition of their IQ. But on the other hand, as I said, they clearly are different than what you would have expected based on their parental IQ status. There are some individuals, and just like with everything, there's quite a range. We have individuals also with an IQ of 120 plus, and also individuals with an IQ of 50. So still quite a range that we were seeing within that group. In addition to looking at something like IQ, if we look at the Social Responsiveness Scale, you can see a similar sort of thing. So again, in this particular case, we've got a low score is good, a high score is worse. So parents, for instance, here's what their SRS scores are here, the siblings, the unaffected siblings here, and then again, in this case, shifted to the right, but again, a standard deviation of about 1.7, almost 2 standard deviations higher in terms of those SRS scales. So again, similar to what Jordan Smoller was talking about, this is something that's really a continuous trait. In some cases, we make an arbitrary sort of line in the sand, and we say that if you're above this threshold, we give you a specific diagnosis. We say that you're affected. But in reality, what this is lots and lots of shades of gray, right? Where there are some additional impairments, some additional challenges that individuals have, but it's not necessarily a black and white diagnosis in terms of that. This is, it seems to be the theme. All of us are using Venn diagrams today, so this is yet another Venn diagram now for the deletion carriers. But to show you that in this particular case, one of the things that you can't fully appreciate from this, but if we looked at DSM diagnoses for our deletion carriers, greater than 95% of them had 1 or more diagnoses. So really, there were very few of them whom you might say are quote unquote normal. They all have challenges in some ways, and in many of them, really, they had multiple challenges. That was actually the theme, is that they had multiple diagnoses, multiple challenges. When you go back and you look in red here, for instance, the ones who met official criteria for ASD, it ended up being, depending on the deletions or duplication carriers, between about 20 and 23% of those individuals who met strict criteria for ASD. So not necessarily everyone met the criteria, but all of them had some features. So they may have had either learning challenges in terms of speech, they may have had some problems with repetitive motion, some problems with need for sameness, but all of them at least had some of those features. Again, when you look at the different disorders, language disorders, certainly a very, very significant impediment, and especially early on for these children. And then in this particular case, we've also shown developmental coordination disorders. But importantly, you can see how many of those children are in the overlapping areas, so that they have multiple diagnoses, multiple challenges. When we looked at the deletion and duplication carriers, deletions on the right over here, you can see that there were many similarities between the two of them. They both seem to have issues in terms of reduction in their full-scale IQ. Again, they had approximately equal frequency in terms of ASD, and certainly they had other frequent diagnoses, although I'll tell you the general impression that we have, and quantitatively as well, is that our deletion carriers are more severely impaired and more severely impacted than our duplication carriers. To be able to show you this for our duplication carriers, what you can see is, again, there are fewer individuals in this Venn diagram with those overlapping diagnoses. There are more individuals who seem to have problems specifically either with coordination or specifically with language or specifically with intellectual disability, but fewer of them have multiple impairments. The thing that was hopeful to us, I think, over time that we were seeing is when we started looking at our longitudinal data, and these are still very early days for us. But this is looking at the children are getting older, so this is looking at their ages in month along the x-axis here, and then what's happening with their verbal IQ scale on the y-axis. And I think we have to be cautious in terms of our interpretation of this, whether or not they're actually changing in terms of their IQ or whether this is our ability to measure it accurately. But clearly what's happening, and I think it's what we see and observe clinically, is that a huge barrier for these kids is around the acquisition of language and to be able to actually be able to get, with the phonological processing disorder, be able to get the words out and to get other people to understand what's going on. Once they do get through that and break through that barrier, it's something that is really, they blossom. They do much better after that. And so this is part of what we're seeing over time, is that as they're acquiring language, as they're getting over that hump, they're doing much better in terms of their IQ measurements. So it's something that I think is some hope that we've given to the families, and also emphasizes how important it is for us us in terms of that early language intervention, being able to get speech therapy early on in these individuals. One of the things that we found first in terms of just asking simple questions about handedness, and then actually as we've been looking at the imaging data, are really, I think, quite fascinating in terms of how the brain is actually organized for these individuals. So this is something called an Edinburgh Handedness Scale. We can do this in terms of measuring how sort of right-handed or left-handed the individual is. In this particular case, in blue, again, are the individuals, the unaffected family members in this case, and you can see that they're largely right-handed, so over to the right here, this scale over to 100, is showing when they're right-handed. When you're around 0, you're basically ambidextrous, or you don't have a specific laterality. And then over to the -100 when you're left-handed. So again, most of our population in this case, as is most of the general population, right-handed. But look how remarkably skewed this population is for the deletion carriers. We have quite a few individuals who are right-handed, but certainly many, many more who are left-handed, and many more who basically haven't decided exactly what they are. And this is despite the fact that they're old enough that they really should have had hand dominance by that point. So realizing just at that simple level that the brain might be organized in a different way. And in fact, as we've mapped the language centers of the brain, we've realized that as well. We don't have nearly the lateralization, the specification that we do in a regular population as we do in terms of when they're processing language. Again, one of the things that we realized early on in this that I don't think we appreciated initially was how frequent seizures were. And so we had initially done just simply a parent report in terms of the frequency either of a single seizure or with epilepsy, and then actually got into much more detailed evaluation of that by literally getting the EEGs, reviewing the EEGs, being able to really document what was going on. And in a good way, we've realized that the frequency of seizures is something that's very frequent, so it's something that families need to be concerned about, but as I'll show you in a second, it's something that declares itself relatively early, and it's something that's amenable to treatment. So it's been actually good news for families in terms of being able to make the diagnosis, make it early, and get treatment for it. When we did this and looked at the age of onset for the seizures, we really saw that almost all the children had had seizures if they were going to have them by the time they were about a year and a half of age, a year to a year and a half. And so the good news is we've had many families, obviously, who have children that are older than that, and we can basically tell them and give them reassurance, if you haven't had a seizure yet, you're going to be okay. Those kids are basically sort of through the period that we would see this on. And as we've done this, as I said, anecdotally we've realized that the seizures were actually quite amenable to treatment, but it was clearly important to be able to make the diagnosis. Interestingly enough, when we went back and looked at the individuals who'd had seizures, looking at, for instance, deletions here on the left, those with seizures or those without seizures, we clearly started seeing a pattern both with deletion and duplication carriers that we had lower full-scale IQs, lower verbal and lower nonverbal IQs across the board for individuals who had seizures. And I don't think that this was simply secondary to seizures or epilepsy or poor control. This really appears to be a primary issue for those individuals. And I'll get back to some of this in a second. In addition, we started realizing, as I said, that the parents were reporting to us that their kids were clumsy, that they were having trouble in terms of coordination. So as we got into the study, we decided we would formally assess this by there's a measure that we have, the movement ABC, which we can actually see how coordinated those children are, being able to toss a ball, catch a ball, their balance, their dexterity, being able to quantify this. And as you can see with our deletion carriers, in almost each one of those domains, they're approximately 1.5 to 2 standard deviations below what the normal population is. Again, the duplication carriers slightly less impaired in terms of that, but being able to quantify what parents were reporting back to us, that there in fact were real problems with coordination and with gross motor issues. Interestingly, again, as we started aggregating the data and looking at who was having trouble in terms of any of those problems with the movement ABC, what we started to realize is that these were clustering together. The kids that had an autism diagnosis were much more likely to have problems in terms of balancing and catching. The individuals, for instance, that were having lower full-scale IQs, similarly having problems in terms of those gross motor movements and coordination. So we were definitely seeing correlations across different manifestations in terms of this. There are a few of you I know in the audience who are geneticists, so one of the tricks we oftentimes use as geneticists for these copy number variants is that although almost everyone has the exact same 600 kilobase deletion or duplication, there are a few individuals who actually have smaller deletions. And so it's something we actually spend quite a bit of time looking for, because as we look at the clinical manifestations for those individuals that have smaller deletions, we can try and determine what are the critical genes within those regions that might be responsible for the different manifestations that we see. This is just simply one example, but this is the full-scale deletion would go from here to here. This individual cuts off another 6 genes within the deletion. And in this particular case, we realized that his phenotype was exactly the same as what we were seeing for the classical deletion, basically reassuring us that the genes within this region for this part of the deletion were not the critical genes that were part of any of the problems in terms of the neurological manifestations. And so we continue to try and identify other similar individuals. I'll switch gears to the neuroimaging, and just to give credit, again, Randy Buckner, who was mentioned earlier today, really has been playing an instrumental role in many of the structural imaging studies that I'll show you, but it's been a large team effort that's contributed to this. One of the things we initially realized, both with the parent report and then reviewing the actual imaging that we did, the MRIs that we did, is that we were seeing problems in terms of Chiari Malformation. So a problem that you can see, this cyst, for instance, back here with this child, but problems oftentimes with the cervical junction, that there were problems in terms of brain malformations, either that had or had not been appreciated. But there was definitely an increased frequency, basically, of a brain birth defect that we were seeing. In addition, although it was not frequent, we sometimes saw polymicrogyria in certain cases, although most of the children, the majority, greater than 80% of the children, actually had structurally normal brains. We went through and we actually quantified this in gory detail, and we had imagers that were blinded, so they didn't know the control siblings, for instance, versus the individuals with the deletion or duplication. And then we started to see subtle things that as we quantified them, we really could see differences. Those things like the corpus callosum being a little bit thinner, again, problems with the craniocervical junction, definitely that we could see in terms of malformations, and things in terms of the white matter volume. Again, remember I had told you about this macrocephaly versus microcephaly, differences grossly that we could see in the white matter volume. We then went on, though, to be able to do very quantifiable volumetrics. So again, we were motivated by this idea that we had already seen that when we just did that simple head circumference measurement, we were seeing differences between the deletions and duplication carriers. So we really wanted to be able to look inside and see if those volumetric changes were really brain volume changes, and was it something that was universal across the entire brain, or were there specific portions of the brain that seemed to have those differences? So we managed to get, in this particular case, 25 deletion carriers to be able to sit still on our scanners long enough to do this, 19 duplication carriers to do the same, and as you'll see in this case, our deletion carriers were largely children and our duplication carriers were largely adults. As we did this, and I'll show you several slides with this, absolutely reproducible, everything that you're going to see, that we always saw consistently the deletion carriers, larger volumes across the board, whereas reciprocally, the duplication carriers, smaller brain volumes across the board. And again, we had this wonderful built-in control where we had family members who could be assessed side by side, or other individuals who are matched by IQ, handedness, as well as gender as controls for this. So again, when we look at this is total brain volume now, deletion carriers higher, duplication carriers smaller. And as you can see from this just gives you a distribution to realize that, these are again contiguous or qualitative traits here, or quantitative traits here, I should say. Our individuals with duplication carriers, you can see their distribution, so this normal distribution shifted up, versus in their cases, their controls. And then in blue, the duplication carriers, again, shifted down. So again, there's overlap between these, but you can, again, just like IQ, just like SRS, you can see a very distinct shift in that distribution. If we started breaking these down by different areas of the brain, we see exactly the same thing, that across all the areas of the brain that we could quantify, we see the exact same pattern. So whether it's the hippocampus, the amygdala, the thalamus, the cerebellum, every single region that we looked at showed very similar and consistent findings. We also wanted to look at the gray matter. So as we did that, we looked at two different things. Again, this is Randy Buckner's work, looking at the thickness. So looking at the gray matter, looking at the cortical thickness and measuring literally that dimension there, as well as the volume and the surface area. And so in the surface area here, literally being able to trace and measuring that surface area of the brain there. What you can see is, again, total volume, that same relationship we saw, deletion carriers larger, duplication carriers smaller. And then if you start getting to cortical thickness, though, quite interesting is that they're exactly matched. So cortical thickness, that thickness of the gray area, exactly the same between the duplication and deletions, but in fact what's different is now the surface area. So the reason why you have a difference in the volume is although that thickness is exactly the same, you have a difference in terms of the surface area, giving you a difference in the volume. And so this, we actually, our current hypothesis is that that copy number variant, therefore, must have very important roles in terms of the brain development. That's a primary thing in terms of the way the brain is initially developing. And that's not so surprising when you also think that, as I said, we had a certain number of children that had literally brain malformations that we could see, Chiari malformations, related things, that at least for many of these individuals, this is probably a developmental process. Obviously, our patients can tell us a lot, but we realize there are some things we're not going to do to our patients. And so one of the things we've built into this study is to have in parallel animal models who can go where their patients can't go, to be able to do more invasive tests, to be able to do things to the mice that we wouldn't do to our kids. So as we've done this, this is Aaliyah Mills' work that's been beautiful work in terms of chromosome engineering in this particular case with the mice. She has made mice that in fact have the entire deletion or duplication region on chromosome 16, as well as now she's making some that have smaller regions with this. And the nice thing is that when you look at her mice in particular, they recapitulate exactly what we're seeing in the individual patients. So again, with the individuals with the deletion, these are mice in this case now, larger brains, whereas those with individuals with the duplication, smaller brains. And that's true, in fact, if you go through all of the different areas of the brain in the mice, just like you do if you see them in the human. So it's been a very nice model for that. In addition, you might not think that this is obvious, but little fish can actually help us out in this particular case. And this is work from Nico Katsanis' lab at Duke. He actually very systematically in the zebrafish went through and basically either increased or decreased the dosage of each of those 29 genes within that interval to try and understand which of those genes might be involved in terms of making a bigger or a smaller head size within the zebrafish. And the zebrafish has lots of advantages to do that. He literally, one of his measurements to doing this was being able to take measurements between the eyes and the zebrafish to try and quantify the volumetrics here. And as he did this, he made a very convincing argument that the gene that's primarily the gene that's responsible for that difference is a gene called KCT13. And reciprocally, again, if you have a deletion or less of that gene, you have a larger head size in the zebrafish. More of it, you have a smaller head size in the zebrafish. So very elegant in terms of being able to use those animal models to understand this better. As we've done this, and as time has rolled on, we also realized, because there is a single gene disorder with a defect in PRRT2 that was associated with epilepsy and a very specific type of movement disorder. And so things have started to fall into place for us in terms of very specific genes within that interval being responsible for certain parts of the neurological or other parts of the phenotype. We have other certain medical problems with vertebral malformations that we think are related to something called TBOC6, but we're starting to now put the pieces together in terms of the puzzle, first starting with the patients and learning from them, as well as I said, bringing other things to bear to take us beyond what the patients can do. I just want to unabashedly advertise, because we have made this intentionally to be a resource to the community. So for any of the researchers that are here, all the data that I've talked about today, we've worked very hard to make freely available. So this is de-identified for any of the families in the audience. So we want to make sure identities are protected within this, but we want to encourage people to think about this and to learn from this. So the sort of great effort that we've taken, for instance, for the imaging, to collect blood samples, to collect cell lines on these individuals are all available so that we can work as a community to try and understand this better. And this includes, for instance, the families who have gone through having skin biopsies done to be able to make fibroblasts, to be able to study these cell lines. Again, all of this, if anyone's interested, go to safari.com and we'll be glad to help you with this. We realize, though, that this is really just the beginning of a journey that we're going on with our families. And to be honest, this is very expensive to be able to do in the way that we did. We also realized that all of our families couldn't come to our clinical sites to participate. It was a long way to go for some of our international families, in fact, impossible to be able to get here to do this. So we wanted to broaden this and make it open to more of our families. So we're actually starting what we call VIP Phase 2. So we're now being able to do much of this online or being able to do it at a distance. So things that people can fill out, families can fill out pen and paper questionnaires, online questionnaires, telephone interviews with us, but be able to do it without having to come in and and see us. This also allows us to be able to collect information longitudinally over time, so we're actually seeing this evolve in different phases. Our cohort at this point is a relatively young, youthful cohort, but there may be different challenges that come up, for instance, in young adulthood and different things that we'll want to see evolve over time. And so we've built that into this. We also realize, though, that we have, to be perfectly blunt about this, some ascertainment bias. From a scientific point of view, we realize that these individuals have come to clinical attention because they had some issues, and so we need to be able to have good comparison groups with them. And we realize while 16P is a good start, it's just the beginning. And so we actually want to be able to expand this, and our most efficient way of being able to expand this to other copy number variants, other single gene disorders, is to basically build a module and clone it. And so that's effectively what we're doing in VIP phase 2. We've extended already with pilot studies to 1Q21, and we'll be able to extend to other studies as well, and being able, we hope, to be able to have a resource that then the research community can use. That is, that will help and facilitate at the Simons Foundation by being able to aggregate some of this data that hopefully others will be able to use much more intelligently and in ways that I haven't even thought of doing yet. So as we've done this, again, the emphasis I want to place is that these are things that are much more complicated, I think, than we realized when we started getting into this. They're not, as Jordan was saying, not these clear set definitions necessarily about DSM diagnoses. These boundaries get a little bit blurred, but clearly the individuals have challenges, challenges that can be overcome to a certain extent, but there are challenges. One of the things that we've seen most commonly, as I said, are language disorders, problems that we're starting to see in terms of lateralization, but we're increasingly seeing that individuals who are challenged are often challenged in multiple dimensions. For whatever reason that it happens, we're more commonly seeing, for instance, seizures or epilepsy going with more severe impairments with gross motor skills, more impairments with intellectual disabilities. So we are seeing people that are multiply challenged in many ways. And as I said, we are starting to see that in terms of the way this is correlating with brain structure, at least, we are starting to see correlations, and hopefully the next time I'll be able to talk, we'll be able to talk even more detail about the function of the brain and some of the and functional studies that we've done as well. So I'll be glad to thank the multiple, we have literally over 100 people that actually contribute to this at many different levels. Again, just a shout out to Ellen Hansen's team here at Children's Hospital. She's done a fabulous job, as well as Randy Buckner, who's been doing the analysis on the volumetric imaging studies. Thanks.